So good morning, everyone. It's my pleasure to have this opportunity to talk about the beneficial microbes and the soil plant microbiomes. And I'm from Plant Pathology Department of Ohio State University. And today I'm going to cover about 3 parts. First, about the healthy plants and soil. Second, I'm going to talk about the soil and the plant-associated microbiomes, and also their relationship with the plant and the soil health. And third, I'm going to talk about the plant disease and its control by beneficial microbes, because I'm a plant pathologist. So we know, first I'm going to talk about the healthy plant and healthy soil. And we know that the plants are very important to us. They provide the basic food, fiber, fuel, and many other materials for us. And they provide us with oxygen for us to breathe for living. And they also help to maintain the sustainable ecology for us, to provide the good environment for us. And but the plant also live in very complicated environment, where they will meet with different kinds of biotic and abiotic stress. For example, in the extreme conditions such as extreme cold, drought, and flooding, and very less nutrient content, and also heavy metals, like a biotic stress, like a passage infection. Plants can get disease, they can even go to death. So the proper condition, like proper temperature, proper sunshine, proper nutrient level are very important. Especially soil are very important for plant to grow. So today I'm going to talk more about the soil that help the plant growth. And the soil is mainly component. It's about 45% of minerals, 25% of iron, 25% of water, and the other 5% is the soil organic matter. And in addition to the soil itself, there are also a lot of living organisms in the soil, such as worms and insects and small animals. But I think the most abundant living organisms are the microorganisms. So these microorganisms are very small. We cannot see them with our naked eyes. We have to use the microscope. And these microbes can include such as the bacteria, fungi, nematodes, and the virus. I will talk a little bit more about it. And so for 1 spoon of soil, it can contain about 100 million to 1 billion microbes. And as I mentioned, these microbes are very important for plants' health and also the soil health. And so the study of the beneficial microbes and also the plant soil microbiomes are very important to help us develop a strategy to get more food to feed the 9 billion people, which will be in like 2050. And before I introduce the plant and soil microbiomes, I also want to introduce a broader conception, which is called Phytobiome. So Phytobiome can include all the organisms that live in, around, and also on the plants. It can include the microbiomes, can include animals, also other plants in the whole system. And I can also include like environmental factors. And today I'm going to focus on the plant and the soil microbiome. And because they are very important for plants and soil. And in 2015, American Phytopathology Society initiated the phytobiome-related research. So the goal is to first to know how the organism associated with the plants can influence, all are influenced by the plants and also the plant associated microbiome, or we see the whole phytobiome. And the other target is to know how the information can be used to improve the crop productivity, quality, and also the safety. And I think the ultimate goal is to, by 2025, we can gain a comprehensive understanding of the phytobiomes, so that to develop a strategy to improve the crop productivity. And as Dr. Aslam just mentioned, that the soil quality and health is very important, and there are three factors like physical, chemical, and biological factors can affect the soil quality and health. And biological factors may be one of the most important factor. And for the healthy soil, I think that is the one that can balance all the three factors. And soil organic matter is one of an important component that can regulate the function of these three components, like biological function, physical function, and also the chemical functions. And also the soil organic matter, they are also from the microorganism, including the plant animal residue that are decomposed by these organism. And as I mentioned before, that there are so many organisms in the soil, and we can group them by size, by species, and also by function. So here, for example, we can group the soil organism into like a living organism and a non-living organism. So virus is a special microorganism because it do not have like a cell. It only has RNA or DNA, but then they can replicate very fast and in the certain condition. And there is living organisms such as a prokaryotic organism, which is just a single cell. Most of them like bacteria, and there's another type of organism, which is a eukaryotic organism, which are higher level, such as they are all like a multicellular organism, such as the fungi, microorganism fungi, such as insects, worms, and the small animals that are inside the soil. And so this is grouped by the size. So we can see the macro or large is bigger than 2 millimeter, such as worms and also the plant roots. And the mass is in between 2 and 0.2 millimeter, so we consider as a medium size. And if the size is smaller than the 0.2 millimeter, then we consider these as a microorganism, such as the bacteria and also the yeast and the virus and so on. So the function of these soil microorganisms are very important. For example, first, they can break down later and recycle nutrients. Second, they can convert the atmosphere nitrogen to the organic form of the nitrogen, and then further to the inorganic form of the nitrogen, so that the plant can take up them as a nutrient. And also they can synthesize certain enzymes, vitamins, hormones, and other important substances for plant growth and also soil health. And they can also alter the soil structure and decompose like a certain weed seeds in the soil. And they can also suppress and feed on some soil pathogens and the pathogenic nematodes. And these pathogenic pathogens and nematodes can reduce a lot of crop yield. And so I think this sentence really describes well about how important of these microorganisms in the soil. So they said that the soil microbiome biomass is the eye of the needle through which all the organic material that enter the soil must pass. So these microorganisms are very, very important. And there are also complex interactions among these soil organisms, and including the microorganisms. So they can be commensalist. For example, they can benefit from one, but do not have a negative effect. And they can also be parasitic. For example, they can benefit from other one, but it's cause some negative effect. And I think that the most interesting one, benefit one is the symbiotic. So they can benefit each other and for their growth and development and health. And the soil-associated microbiome generally include the bacteria, actomycete, fungi, and virus. So these microbes, they can interact with each other. They can also function together with each other. And they can even have interaction with other organisms in the soil and also include the plants. So I will talk more about how these microbes involve in the interactions among microbes of plant, and plant with soil. So it is very complex interactions. So this picture, I just want to emphasize that these beneficial microbes really need a function together. So it is not just a single microbes exists in the soil. They have to function together to play their roles. So next I'm going to talk about the plant-associated microbiome. Just I already mentioned Phytobiome important components, soil and microbes. Now I'm going to move to the plant and plant-associated microbiome. So plant-associated microbiome are the microorganisms that are on the plant, inside the plant, and around the plants. So the soil microbiome are very important, just like our human microbiome. Like our human also, we also have like a microbiome in our gut, in our skin. They can also play very important roles in our development and also health. And so because the plant-associated microbiome is so important, so people also consider the plant microbiome as the secondary genome of the plant. And the plants, as I just mentioned, these microbes can be on the surface of the plant, which we call it the phyllosphere microorganism. And they can also be around the root, rhizosphere. And they can also be inside the plant. They can be inside the plant through wounding, by stomata, and some endophytes. They can stay their whole life cycle in the plants. Some can just live a certain life cycle in the plants. And generally, we think that endophytes can set up a more close relationship with plant because they have to manipulate the plant immune system to help them enter the plants, develop in the plants, and in many cases, the endophytes can benefit the plants, and also they also get the benefits from plants. So most of the cases, they can be symbiotic relationship. And the approach to study the plant-associated microbiome also developed quite fast with the last 10 years. And I want to introduce that there are about 99% of the microbes in the soil cannot be cultured. Actually, only 1% can be cultured. So But although there's only 1% can be cultured, they can also be very important. And we study for example, we can isolate these microbes from the plants. We can culture them on a certain medium, and we can extract DNA by PCR sequencing. We can identify them, and we can study the produce and metabolize. We can inoculate them on plants, and to see if they can promote plant growth. We can benefit the plant in a certain way, and we can also even make the community, synthetic community. For example, we mix several microbes together to see if they can increase their beneficial effects. And for the unculturable microbes, they are developed an omics approach. For example, the metagenomic, metatranscriptomics, metabolomics, which is omics approach. We can also study these community in the DNA, RNA, protein, and metabolite level. And plants interact with the soil social microbiome mainly through their roots. And the plants actually can release 30% of their photosynthesis products to the soil, which contain mainly the organic acid and also a complex of phenolic compounds. And these acids and the phenolic compounds themselves are very important to benefit the plants directly. For example, these organic acids can mobilize the plant nutrients, and the phenolics can kill the soil pathogens. And also, the secretion from the root also very important. They are the compounds that sometimes they can attract certain microbes to the plants. So plant looks like it has the ability to attract certain beneficial microbes to help them grow. So they are very smart actually. And also the study showed that the rhizosphere soil is really different from the book soil. So the plant can play important roles to recruit these microbes. Not only microbes affect plant, the plant can also play roles actively recruit these microbes. And plant and the microbes, they generally use certain signals to communicate to each other. For example, they can produce certain metabolites. And also the microbes and the microbes, they can also communicate with each other through certain metabolites, small molecules. And then we can also communicate with plant, not only in the root, but also like the upground tissue. And this picture shows the potential of the rice endophytes that can play roles in these plant microbe beneficial interaction. For example, it can help the endophytes colonize to the plants, can help the endophytes to live and develop in the plants, and they can also help the plant to get more nutrient, and also increase plant resistance to biotic and abiotic stress. And they can also promote plant growth. For example, they can produce a certain plant hormone, or they can help the plant to do nitrogen fixation, or to taking up more nutrients. So there are very complex interactions among the plant, soil, and also microbes. And as I want to emphasize again, that all these have to work together as community and to function together. So there are several questions that we needed to answer from the Phytobiome study. For example, what is the genetic linkage that connect the Phytobiome components? Can we breed plants that select for the beneficial communities? Another question, what constitutes a healthy phytobiome? And can we develop biological and predictors of crop and soil health? And many other questions we want to answer is, can the phytobiome be managed to maintain the soil health in an environment-friendly manner? And can we rebuild the depleted soil by restore the phytobiome? So these are the questions and the research that is needed to be further investigated. And next, I'm going to talk about the plant disease and also plant disease control by beneficial microbes. And as I mentioned, that plant pathogen can cause very severe plant disease symptom and reduce the yield. And for example, in America, every year we can get about $4 billion loss caused by these pathogens. And about 90% of these pathogens live in the soil. And there are conventional approach to control plant disease, such as by the chemical application. But it can also cause problem, can cause long residue, give pollution to the environment. So today I'm going to talk more about the non-chemical control for plant disease. For example, we can use crop rotation, we can use plant spacing, tillage, soil solarization, plant host native defense. And today I'm going to talk more about the biocontrol agent, suppressive soil, and also the induced defense that can be induced by the beneficial microbes. So this is the field of the wheat field, and the X is the years that the wheat grow, and the Y is the disease severity. And we can see at the very beginning, after by infection with this fungi, naturally, the wheat got disease. We can see very severe symptom. And this field, people didn't do anything on this field. They didn't do rotation or tillage. They just live there. So after 5 to 6 years, and people found that the plants can be very resistant in the field. So this is very interesting phenomenon. And so the researcher found that in the natural condition, actually with the pressure of the pathogen, the microbiome in the soil can be regulated. So the composition of abundance can be changed. And they found that Pseudomonas strain become very, very abundant in the community. And this bacterial strain, they can produce antibiotic. And this antibiotic can kill the pathogen. So under the pathogen pressure, after 5 to 6 years, the soil reprogramming its associated microbiome and produce more microbes that can kill this pathogen. So they can become like a disease suppressive soil. And actually, in natural condition, there are many cases the soil can have the opportunity to become disease suppressive soil. And they also have like physical attributes, chemical attributes, of course, also microbial activity. And also, the transfer, people found that if we transfer these disease-suppressive soil to other soil, mix them together, actually they can also cause the other soil confer disease resistance, so it can be transferred. And also they found that if they pasteurize these soil, they can lose the activity. So which means that the activity of these microbes, their living well is very important to confer the disease suppressive. And I think that the surface cell not only can suppress the disease pathogens, but can also suppress, for example, the wheat seeds and the arthropods, and also some other insects. And here I also want to introduce one of very important beneficial microbes cause disease resistance, which is called induced systemic resistance. So there are certain microbes, they can live in the soil. We call it plant growth promoting bacteria, PGPR. They live in the soil and they can interact with the plant through the roots, for example. And there are many beneficial microbes can inhibit the pathogen growth. For example, they can produce antibiotics, and they can compete the nutrients or living space with the pathogen. And but there are also some beneficial microbes. They can also trigger the systemic immunity, just like the vaccine of human, right? They can trigger our human immunity. So these beneficial microbes can trigger plant immunity. So the plant, after these beneficial microbes interact with plants, they can trigger plant immunity. So the plant can confer resistance to pathogen infection, to pest infection. And generally, this mechanism is related with the production of certain plant hormones, such as jasmonic acid and ethylene. But I think the further mechanisms still need to be investigated. There's still a lot of unknown mechanism existing there. And this figure shows how the soil microbial plant soil organism, how they interact with the plant root. So this is soil organism and microorganism. So the plant root can secrete certain certain compounds, small molecules, which can attract these microbes to the surface. And also these microbes, they stay at the surface, so we call it a phytosphere. And also these microbes, they can accumulate more and more and more and more, and then they can actually help the plants to inhibit a certain pathogen. For example, they can produce antibiotics, or they can compete the nutrient with pathogens, so repress the pathogen and increase the plant disease resistance. And these are the microbes that can go inside the plants, which we call the endophytes. So most of them can be very beneficial to the plants, but not all of them. So this is the figure shows that after beneficial microbes treatment, we can see it can really increase the lateral root growth of the biopsy plants. And this is the seedling under microscope. We can see a lot of labeled microbes existing in the plant root. And this is a table of the beneficial bacterial strain, PGPR, that has been identified and tested to promote plant growth and inhibit pathogen growth. So we can see these are from different plants, from different pathogens. They can actually inhibit both fungal pathogen, bacterial pathogen, and EV virus. So they test on different plants also. And actually, many of these beneficial microbes can not only be used as a biocontrol agent, they can also use developed for like a biofertilizer or biostimulants. And like this strain, WCS417 of Pseudomonas has been commercialized by the Bayer crop stands. And this is another picture shows that after this bacillus bacterial strain treatment, it can inhibit the virus growth. We can see here a lot of virus grow in the tomato plants, but not here. In addition to the PGPR, the plant growth protein bacteria, there are also some other fungi can be involved in this beneficial interaction. For example, the McCarthy fungi, They can also play very important roles in inhibiting plant pathogen as a biocontrol, can be used as biofertilizer, and biostimulants, and for soil phytoremediation. So there are very complex interactions existing in the phytobiome, and there are interactions between plant, microbes, and microbes with the soil with plants. So very complex, and so I just want to emphasize again, they have to function together. So beneficial microbes can affect plant can also affect microbes, and also as to the soil. So to make a conclusion, I want to say that the study of the mechanism of regulation of the whole phytobiome, especially the plant and the soil-associated microbiome, are very important for us to develop further strategy to improve the soil and plant health, to increase plant yield, so that feed 9 billion people in 2050.